Right, okay. I first met Martin Brasier on a field trip in 1992 in China. A fantastic experience going up the Yangtze gorges at that time, and we learned a lot of geology and paleobiology as well, and generally inspired. Martin has been very supportive of my work over the years, and our research paths have crossed repeatedly. I know that Martin is interested in the evolution of eukaryotes, and that's what I'd like to talk about today, if I might. How does this work? Won't bother. What you're looking at here is a timeline. And we're curious about this biosphere that we live in today, which is dominated by eukaryotic organisms. It's not to say that there's only eukaryotic organisms, but they structure the biosphere in which we live. The question is, was it always this way? How far back in time can we track this eukaryotic structured biosphere? Well, we've got a fossil record, and the Phanerozoic fossil record is dominated, certainly conspicuous by its presence of eukaryotic organisms of one sort or another. The question is how far back in the record we can track that eukaryotic dominance, eukaryotic signature. One suspects not all the way back. There's going to be some sort of development of that eukaryotic biosphere. We can go back to the beginning of the Proterozoic, which is broadly coincident with a significant rise in atmospheric oxygen, the great oxygenation event. So at least by the beginning of the Proterozoic, 2,500 odd million years ago or so, we've got a diversified prokaryotic biosphere with things like cyanobacteria in it. We're interested in the origin of eukaryotes. Where do they come from? Well, eukaryotes, pretty much everyone agrees, evolved from something with a prokaryotic grade of organization. And we're in the business then of understanding how this rather remarkable and exotic kind of creature here evolved from something substantially more simple. There's a fundamental change in grade of organization. There's a fundamental change in the level of the type of individuality. This is a different kind of thing altogether, right? It's the deepest, greatest constitutional divide in biology, at least arguably so. And not surprisingly, it attracts all sorts of interest in how it came to be, getting from that prokaryotic to eukaryotic grade of organization. There are some really bizarre ideas out there, let me tell you. But before I do, let me just apologize to Nick Lane beforehand. Nick Lane is one of the more clever and imaginative evolutionary cell biologists around, but you know, he's got crackpot ideas about how things are worked. Here's the archaea. We know that eukaryotes have a component of archaeal and bacterial inputs. So here's the bacteria and the archaea, and here's the eukaryotes deriving therefrom. We recognize that there's a huge genetic and substantial distance between these prokaryotic groups and the eukaryotes. But these guys perhaps looking a little bit too closely at extant organisms, find it almost impossible to see how you can evolve one from the other in any sort of conventional sort of way. And there's just way too much of it. There's all sorts of hopeful monsters out there. The argument is that there's a big bang, and eukaryotes just appeared on the scene. It's crazy stuff, and I don't think it's the right way of looking at all at the question to hand. As paleontologists, I think we have this sense of deep time and recognizing that there's all sorts of extinct creatures back there, and those extinct creatures are very important. They tell us how stuff is assembled over evolutionary time, and I think this is much more the right kind of way of thinking about things, and this has been promoted by all sorts of paleobiologists, Dick Jeffries, Bruce Runegar, Graham Budd, is impressed upon us that we need to think about the distinction between stem groups and crown groups. Crown groups is defined, originates from a last common ancestor, but the total group derives from a first common ancestor. And the point is there's lots and lots and lots and lots of extinction. It's all that distinction that gives us the phylogenetic divides, the divides that define a lot of the major groups. So here is a schematic cladogram looking at the big picture. And right down here we've got Luca. This is the last universal common ancestor of all life. This is an interesting diagram. It's reflecting the current views coming out of Martin Brasier's lab, and I think is fairly widely agreed on, Williams et al. Most of you will be under the impression that there are three major domains of life, right? The bacteria, the archaea, and the eukaryotes. It turns out that that's not the case. What you see here is there's just two domains of life here. There's bacteria and there's archaea, right? The eukaryotes have ended up here. Here's the last eukaryotic common ancestor, Leca. And that's something that I'm particularly interested in resolving in time. But what you'll notice is that Leca is most closely related to a subset of other archaebacteria, namely the so-called TAC archaebacteria. These guys, these guys, these guys, and these guys. The most recent molecular phylogenetic analyses put eukaryotes as sister group to the TAC archaebacteria, to the exclusion, that's no good me pointing at the screen, to the exclusion of the eukaryote. So that's of passing interest. The archaebacteria are a paraphyletic group out of which emerges the eukaryotes. So there's this last eukaryotic common ancestor here. Here's Leca. Here's the first eukaryotic common ancestor. The way to think about how you end up with that last common ancestor is the accretion of the particular characters along that stem lineage until you get the full complement up in Leca. And there's a list of all the, it's just a universe apart, those things. But somehow or other, I think we have to sit down and worry about how you get from this prokaryotic grade creature down here to this eukaryotic grade one up here. Here's an interesting little tidbit to this tale. It comes from the Pilbara, from the early Archean in Western Australia. Evidence for fluid inclusions for microbial methanogenesis in the early Archean era. Methanogens belong exclusively to the Euryarchaeota. We've got evidence, it's not absolutely watertight evidence, it's pretty good evidence for Euryarchaeota at 3.5 billion years ago. That implies a sister group of the Euryarchaeota was also present at that time. That implies that total group eukaryotes were present at 3.5 billion years ago. You heard it here first. There's 3.5 billion year old eukaryotes in the record. There's good evidence for it. But it's total group eukaryotes. We're talking about total group. We're talking about Feca rather than Leca. So let's look more closely at Leca. Here's the last eukaryotic common ancestor, and it's divided into a number of major eukaryotic lineages. And there you see your favorite ones. The excavates seem to be different from all the others. And then this is essentially the biconts and the uniconts in Cavalier-Smith world. And up there at the top will be metazoans, fungi, embryophytes, and so on. Really? Wow. Is that 5 plus 5?
Then you reach it into your question.
Yeah. But I can pull a Graham Shields, though. We want to know what the nature of Leca was. The molecular guys have done a lot of work on this front. Leca was a really complex. It had all the basic machinery of off-the-shelf eukaryote from the modern world. It was really complex. It was a flagellated amoeboid. That's useful to know in passing. How are we going to go? Here's the question I want to resolve. I want to know when crown group eukaryotes turned up on the scene, okay? With a full complement of stuff capable of doing what crown group eukaryotes are capable of doing, whatever that is. This is going to be a difficult thing to capture. How about using biomarkers to capture them? Eukaryotic steranes, 27, 29, steranes, sterols in particular. You heard it here, and I apologize to Joachim Brox and biomarker workers everywhere. This doesn't help one whit in understanding this node here. Those steranes could have happened anywhere along that stem lineage. And you're right back at 3 point. It's not going to help us one whit. We got to, in other words, the only thing left, we got to go back to body fossils. Body fossils, it's a ghastly record, but we're forced to use them, and it's more interesting than you imagine. I don't have time to go through all of these things. I'll just point out that there are some pretty good body fossils out there that can help us out. And in particular, so here's the question. What I'm curious about is crown group eukaryotes, everything above LECA. How do I know I'm in the crown group as opposed to in the stem group? One way is finding members of an extant subgroup of eukaryotes. So I won't go into it. This is Bangiomorpha. It is as good as it gets, and it's really good. This is a bona fide Bangiophyte red alga at more or less 1,200 million years ago, okay? We've got crown group eukaryotes at 1,200 million years. Here is a Proterocladus. It's claimed to be a siphonocladalian chlorophyte. It looks like it. This thing is not quite so written in stone. It's morphologically very simple. You have to acknowledge that this form could well have been converged upon by some other simple eukaryotic filamentous kind of beast that wasn't a chlorophyte at all. So I think this is less absolutely certain. Vase-shaped microfossils, probably testate amoebae, but it's a very simple form. You might wake up at night worrying just a little bit that this is a Conway Morris world, and this form might have been converged upon. These are a couple of forms here. You have to worry a little bit. Here's a well-known form that some of us worry about, Paleovoucheria, said to be an ancient Voucheriacean xanthophyte alga. It looks like what you're looking at right there looks like a modern Voucheria, but this is a really fascinating specimen here that Tamara Herman described a couple of years ago. She gave it a different name, Amonema, and it is exotically different, but you know what? It's just a more complete articulated specimen of Paleovoucheria, and what it does, it reveals how misled we were in looking at bits and pieces of this thing and viewing them as Voucheriaceans. This thing is definitely eukaryotic, I would say, but it's sure as hell not a Voucheriacean. I described this thing here as a proterozoic, a probable proterozoic fungus a number of years ago. It's probably not. It's not good enough, quite frankly. We've got to be more serious about throwing names around, I think. So let's come back to this. I've got 0 time left, right? Okay, come back. But remember now, the question isn't, can we find proterozoic fungi or chlorophytes? The question is, can we find crown group eukaryotes? And the question is, are these things about which we don't know very much crown group eukaryotes, or are they stem group eukaryotes? Are they pre or post LECA, right? This thing, I'm betting this is a crown group eukaryote. It's found in the Ediacaran. Just because I can't really say what it is, doesn't really bear on the matter. I'd say the same thing about that, but it's how do we go about interpreting problematic things that look eukaryotic that don't fall into extant subgroups, and saying whether or not they are bona fide crown group eukaryotes. Well, here's my list of criteria. One, they've got to be really complex. Morphologically, ontogenetically, however you look at it, they've got to be complex. Stratigraphic continuity is a useful measure if you've got a complete record of crown group eukaryotes going all the way back. Then a problematic thing may not be so problematic after all. Co-occurrences with an extant subgroup. Okay, so this ediacaran thing co-occurs with extant subgroup. It's fine. I'm not going to bung this down to pre-Leca interval just because I don't recognize what it is. I think that's fine there. We've got testate amoebae here. These scale microfossils, they're really, really exotic. But I don't know what the hell they are, but they co-occur with crown group eukaryotes. They're fine in my books, right? In the early Neoproterozoic to late Mesoproterozoic, we've got a bunch of stuff. Don't know what they are. They're acritarchs, but they co-occur with things that we do recognize. So, and that's not a watertight case. Just because I've got crown group eukaryote here doesn't mean these things necessarily are, but I think that's we've got to make some compromises somewhere. I think that's good enough. These are crown group eukaryotes. Going back into the Mesoproterozoic, the middle to early Mesoproterozoic, there's this thing called Tapania. It's got stratigraphic continuity, the same sort of grade of organization, same kind of complexity, going back. There's no abrupt transition there. I'm happy to say these things are crown group eukaryotes, even though I don't have any hand on heart crown group eukaryotes co-occurring at this time. We've lost anything that's immediately recognizable. Never mind that, though. We can go back to the late Paleoproterozoic. There's a couple of really, there's a really interesting new date up there. This date has been around for a while, but they look like they're very good dates. Tapania, it's exactly the same species of Tapania, extends into the late Paleoproterozoic, 1600, 1630 million years. If you want to be an ultra-skeptic, fine, because there's no co-occurrence of recognizable extant subgroup crown group eukaryotes in there. But I'm pretty happy with that. I'm pretty happy that we've got crown group eukaryotes in the late Paleoproterozoic. That's complemented by another thing, which is very complex in quite a different way. So as I say, I'm happy to conclude that these things are crown group eukaryotes. Here's one more little tidbit. Not only are these really complex, they have cell walls. Cell walls is what allows them to be preserved. Because if you don't have a cell wall, you're paleontologically invisible in this business until you get Doshanto type preservation, which doesn't exist until you get to the Doshanto times. Leca was a very complex thing, but it was a naked flagellate amoeboid. It is paleontologically invisible, okay? You'll never ever see that thing. It's only, I think it's fair to say, well, you only get eukaryotic cell walls, so these things only start appearing in the fossil record up in the higher reaches of this tree. These things have exotic and exotically different types of cell walls. These guys are crown group eukaryotes. 1630 million years is a minimum paleontological date. So let's look at our timeline again. Here we are. What about molecular clocks? Molecular clocks are all over the page. There's a bunch of popular ones just come out up here. This is a sensible looking one. To my eye, this is a sensible looking one as well. Namely, you can pick and choose in the molecular clock business. This is billion-year extrapolation going on. They're meaningless, effectively. But the body fossil record rules out these guys. I'm telling you, these ones are wrong, right? And know who's done it? The body fossil record did it. I kind of like this one back here, because you know what? I kind of think that major innovations in evolution have something to do with how the planet works. And there's some nice stories. I've got one of them Bhattacharya has another of how and why eukaryotic LECA, the evolution of LECA, might correspond to the great oxygenation event. And make them up as you go along. So let's just say, okay, what about that biomarker record? Biomarker record, according to Jochen Brox, the first time you get hand on your heart, abundance, isomer, all those sort of necessary signals that you need to see, around about 750 million years, you start seeing a real presence of these eukaryotic things. And you go from effectively a cyanobacterially dominated biological pump to a eukaryotic one, around about 750. Yep, we're here. Not only that, around about 750, in fact, right on 750, you get a whole new range of seriously armored, large, biomineralized eukaryotic microfossils turning out. That kind of, it's interesting. What we've got then is 1 billion or so years from when LECA first arrived until eukaryotes are putting in any sort of serious ecological presence. Why is that? I'll tell you, the Butterfield hypothesis is that these guys are the most ecologically insulting component to ecology that the planet has ever seen. And there are, for what it's worth, not very much, molecular clocks, put them turning up on the scene around about 750 million years. It makes a nice kind of story. They're paleontologically rather indistinct, but it's only a relatively little while before you tip into snowball Earth and then tip into Cambrian explosion. It makes a nice big picture story. Here's a wrap up. Here are the conclusions. The Fekete-LECA transition is paleontologically invisible. We're never going to see it. We've got to make it up. Body fossil record is the best of a bad lot. And it is a really bad lot, but body fossils are really the way to go. Morphologically complex fossils, the kind we were looking at, are crown group eukaryotes. I think you can take that probably to the bank. Total group eukaryotes first appeared in the early Archean. Crown group eukaryotes first appeared in the Paleoproterozoic. The assembly of LECA took a couple billion years. Martin wrote a paper on how long it took to assemble eukaryotes. Maybe there's something to talk about there. What's interesting is it was another billion years before eukaryotes with this enormous potential to do stuff. It took another billion years before they actually did that stuff. And what was required, I think, is it's not just potential. You have to have the opportunity. You have to have the drivers. I think why it took such a long time. It took a long time to invent that eukaryotic grade of organization. And then those animals are enormously powerful drivers, almost everything. It took a long, long time to invent those by accident because those things are extraordinarily complex. Once you trip over that unique combination, away you go, and you've invented the eukaryotic biosphere. Thanks very much.